Okay, now we are moving on to a group we call protists. And very quickly, you're going to learn that protists are not prokaryotes. These are the tiny eukaryotes.
Up until now, we have been talking about prokaryotic cells, so those that fall in the domain of bacteria and archaea. Now we are going to move on to eukaryotic cells. And it is thought that, according to the theory of macroevolution, that the eukaryotic cells diverged from the domain archaea about 3 billion years ago. So that's according to that theory. If you'll remember correctly, there were some distinguishing features that make eukaryotic cells different from prokaryotic cells. So let's just review this. You should already have a copy of this from when we were comparing the three domains. So the types of cells, again, we're talking about eukaryotic cells in the domain eukarya instead of prokaryotic cells. And I don't know why my little eukaryotic peptidoglycan cell wall doesn't fill in at the very beginning. But the peptidoglycan cell walls are only present in the bacteria prokaryotic cells, so we don't have any of that in our eukaryotic cells. But it's much more organized than the prokaryotic cells. It has membrane-bound organelles, larger ribosomes, and it now has introns. So now, like I said, introns are the portion of the DNA that doesn't get transcribed, if you'll remember that. And I personally just think that we don't know what it does yet, because it has been called the trash DNA for a long time because it gets cut out. But I can't imagine that if we have decided that eukaryotic cells are more advanced, why they would have them, and prokaryotic cells mostly don't. So they must serve some function, but it's not clear just what that is yet. So our eukaryotic cells are found in the kingdom Protista, fungi, plants, and animals. Okay, there are 4 features of eukaryotic cells that we are going to go into, and they're required in order to be categorized as eukaryotic cells. The first is the cytoskeleton. Just like we have a skeleton, every individual cell has a cytoskeleton that helps to give it support. And it gives it a particular shape, and it can also help in movement. So eukaryotic cells contain a cytoskeleton. The cytoskeleton of a cell, if you look up here in the left corner, it is stained green for actin, this right here. And actin is just one type of protein that helps to make up part of the cytoskeleton. So the cytoskeleton has 3 different components to it. The first is microtubules. And microtubules just really act like scaffolding. It helps to shape the cell. It's also like, if you think about train tracks and how things get moved from one end of the cell to another, those are on microtubules. And we'll also see with, like, for the flagella, they're arranged in certain patterns that allow for movement. The second component of the cytoskeleton is microfilaments. That's made up of actin. That's what's over here in this picture on the left. And it's considered a contractile protein. So actin and myosin work together to contract and expand and allow for cell movement. And it's also responsible for cytokinesis. The third component is the intermediate filaments. And those are just additional proteins that add additional structural strength to the cell. I've added this link below so you can take a look at what we call cytoplasmic streaming. And this is just a great video that shows that, and it's kind of hard for me to describe it to you, but basically the cytoplasm within each cell continues to move, and it almost looks like blood flow within a human, if that gives you an image. But I would like you to go over to that YouTube site and just take a look at it, so that you know what the cytoskeleton is responsible for. Okay, the second characteristic of a eukaryotic cell is that it undergoes mitosis. So it's a much more organized way to distribute genetic material. If you remember, with our prokaryotes, we had binary fission. So this is much more complex. Okay, the third component of a eukaryotic cell, remember we mentioned that there are membrane-bound organelles. And an example of that would be the mitochondria, the site for cellular respiration within a cell. And then the fourth component would be sexual reproduction in some eukaryotic cells. Some are still asexual, but we have the introduction of sexual reproduction for the exchange of genetic material. And with that, there are 3 different types of life cycles. And just kind of jogging your memory, do you remember what haploid and diploid mean? Haploid being 1 copy of the genetic material, and diploid meaning 2 copies of the genetic material. So the first type of life cycle is what we call a diploid life cycle. That means that the organism spends most of its time in the diploid phase, and we as humans are a diploid dominant life cycle. So for us, that means pretty much all of our lifetime is spent up here in this 2N phase. The only time that we have a haploid portion of our life cycle is when we produce egg and sperm. And so that means that we are a diploid dominant life cycle. The second type of life cycle is what we call the haploid life cycle. And just as you would guess, organisms live most of their lives in the haploid phase. And we see that in the kingdom fungi, and we'll get to that later. And so if you are looking down here at this life cycle, you will notice something a little bit different that we'll get into. But the big thing to notice is that this entire portion is the haploid life cycle. Fungi do something a little bit different, where they do have a phase called the dikaryotic phase. That means they have, that's before their nuclei fuse. So they have a cytoplasm with individual copies of 2 different genetic sets. So that's the dikaryotic phase. And then they spend a short time in the diploid phase right here, but the majority of their life cycle is spent in the haploid phase. So that's the second type of life cycle. The third type of life cycle that we see is what's called the alternation of generations. And this means that the organism lives part of its life as haploid and part of its life as diploid, and that both of those phases are multicellular. So if you remember, in the diploid dominant life cycle that we live, our multicellular being is in the diploid phase. The only time we have a haploid phase is single cellular, so the single egg or the single sperm. So plants are unique in that they have an alternation of generation, which means they live in both the haploid stage and the diploid stage, both in multicellular organisms. So the fun thing about the protists that we're going to be talking about, which are the simplest eukaryotes, is that they can display they can be found in any of those 3 different types of life cycles. Okay, so now we know we're in the eukaryotic domain. We've discussed the characteristics of eukaryotic cells. So now let's get to protists. What are protists? The first thing that I'm going to say over and over again is protists are not prokaryotes. Okay, because oftentimes we see the word protist and we think, oh, then they're simple, they're prokaryotic. No, we just spent the last several slides going through, they are eukaryotic. So, but they are the simplest but most diverse eukaryotes. They can be anything from single-celled to colonial, or a simple multicellular. Typically, they are asexual reproducers, but they can produce sexually as well. They form spores or cysts in order to reproduce. Many of them are what make up the plankton within a marine environment. If you don't know what plankton is, it's that microorganisms that are suspended in the water, typically at the top layer of the water, and they're usually autotrophs. And so if in this picture it says microscopic algae, algae is more of like a common name for autotrophic protists, and we're going to get into that as well. Another important thing is that protists do not show tissue differentiation, so they aren't organized to the tissue level yet. So how is it thought that protists came to be? There is a theory that was proposed by Lynn Margulis in the 1970s called the endosymbiotic theory. And this was her way of explaining how the simple eukaryotic cells came to be. So what the endosymbiotic hypothesis is that there was one particular prokaryotic cell that engulfed a purple aerobic bacteria and managed to engulf it and keep it and not break it down, but begin to use it to produce energy, and that became the mitochondria. Similarly, then after that same type of cell then engulfed a cyanobacteria, which is an autotrophic bacteria, and those properties were kept as well, and that became the chloroplast. So her reasoning for this was specifically that the mitochondria and the chloroplasts have two membranes, so that would pretty much jive with the idea that it was engulfed. So you would have the original purple aerobic bacteria's membrane and then the vesicle membrane that engulfed it. So that would be the two membranes. And then mitochondria and chloroplasts both have their own ribosomes, their own DNA, and they can replicate by themselves. And they're about the same size and shape of other prokaryotic cells. So that is the theory of endosymbiosis, and that was proposed by Lynn Margulis. So I would like you to take a minute, again, I know today you're hopping over to lots of other pages, but to go take a look at this video. It explains the endosymbiotic theory very well, gives you a visual with that. So I want you to take a moment to look at that.
Now that you've had the chance to look at the videos and you've seen the supporting evidence for the theory of endosymbiosis, I want you to start to think about reasons that theory might be incorrect. It's a relatively new theory since it was just introduced in the late 60s, early 70s. And so in my class, I always want you to be thinking critically and not just accepting whatever you're told. So I would like you to go over, this will be one of your additional assignments, and it will be on your site for you to submit just really a quick paragraph or two that is some evidence that you can think of just based on what you know as to why this theory might be incorrect. I don't expect you to have all the answers, so there's really no right or wrong. I just want you to be able to support what you're saying in a logical way.
So looking on this particular chart from left to right is time. So we start with the first cells basically in this far left, and these would be what we call LUCA, remember when we first talked about that? That would be the last universal common ancestor. And then of course, the breakout between archaea and bacteria. And if you look sort of in the center section, this is where eukarya is going to branch off. And so our eukaryotic cells flowing this way, this is where our aerobic bacteria was engulfed and thought to be the mitochondria, and then later we would have the engulfment of the chloroplasts, or the cyanobacteria that produces a chloroplast, so that leads to our autotrophic eukaryotic cells. Okay, so we have definitely established what eukaryotic cells are, the thoughts of where they may have come from. So now let's get back to Protista. Our protists, remember, are the simplest of the eukaryotic organisms, and the phylogeny seems to be pretty complicated, so therefore they're seen all throughout the various stages, excuse me, all the different categories. Oh, yes, I should say that. We used to have a kingdom called Monera, and that was all the protists. So they were just kind of thrown into this junk drawer. Now, based on phylogeny, we've put those simple protists in the various categories all throughout. Over here on the right, if you can look where we have all the different eukarya, rhizaria, and just and amoebas, all those kind of things. So it's not in just one kingdom anymore. It's spread throughout the other kingdoms.
So because protists are spread throughout a variety of kingdoms, and we still want to look at them in the sense of their nutritional methods, scientists came up with an extra category in the hierarchy. If you'll remember, we talked about everything from domain to species, where domain was the most inclusive and species was the most exclusive. So scientists decided to add an extra category in there called a supergroup. And the supergroup is below domain but above kingdom. And if you take a look over here on the left, this chart kind of tells you what supergroups there are and the various protists that are seen in it. So we have the archaeoplastids, the chromoviolatas, the excavates, the amoebozoans, which typically hold your amoebas, your rhizaria, and the apostacanthes. So we're going to be looking at these different supergroups, but in a more broad sense. So we're going to take these protists and look at them based on their nutritional modes, and then I'll tell you which of these supergroups that falls under.
Like I said, protists are found in all 6 of the supergroups. Today we are going to start talking about the first 2 supergroups, the archaeoplastids and the chromoviolatas. And so we're going to be looking at red and green algae. We're not going to look at land plants because those are not protists. And we're going to look at the brown and golden brown algae, as well as molds, diatoms, dinoflagellates, and the alveolates. So let's go on to the next slide. Okay, this is how we're going to break it down for our simplicity to look at it. We have the plant-like protists that are photoautotrophs. We have animal-like protists that are heterotrophs, that are also called protozoa. And then there are fungal-like protists that are saprophytic. And so today we're starting with the plant-like photoautotrophs, and those are mostly algae, also diatoms and dinoflagellates. So just a terminology for you to remember, phycology is the study of algae, so those plant-like protists. Okay. So our photoautotrophs, like we've talked about before, undergo photosynthesis. So why are algae not plants? Some people would look at them and go, oh yeah, those are green plants. Okay, they are not. And the reason why is because they don't have differentiated tissue. So in order to be a plant, by definition, you have to have differentiated tissue. So roots, leaves, and stems, those are all differentiated tissue that contain xylem and phloem, which we are going to talk so much about when we get to plants. I can't wait. But for now, just to know that algae do not have true roots or true leaves or true stems because they don't have that vascular tissue. So therefore, they are not plants. So our plant-like protists, like we said, they undergo photosynthesis, so they're photoautotrophs. And the green algae are in the supergroup archaeoplastids. Archaeoplastids are so named because they contain plastids that are thought to have been absorbed by absorbing a cyanobacteria. So the general characteristics of our archaeoplastids are that they contain chlorophyll A and B, just like plants do. They store food as starch, and they contain cellulose in the cell walls. And for those three characteristics, those are the reasons that the archaeoplastids are thought to be the most closely related to land plants. Okay, but our archaeoplastids are broken down into two main groups. So our green algae falls under chlorophytes and charophytes. So chlorophytes, in favorable conditions, undergo asexual reproduction, and they often have a flagella. And so some examples of this that you will see in lab are Chlamydomonas and Volvox. On the other end of the spectrum are the charophytes. These are the filamentous algae, and they undergo sexual reproduction. And this is the category of archaeoplastids that's most closely related to land plants. And two examples of that you're seeing in lab are Spirogyra and Chara.
So green algae have four structure types, so four different ways that you see them living. The first that we're going to talk about is that they can be unicellular, which means they're just comprised of one cell. And our typical example of that is what we call Chlamydomonas. So this is what it looks like. Get all this in here. So Chlamydomonas lives as a haploid dominant organism. So that, remember, that means one copy of the DNA. And it's a chlorophyte, as we mentioned, and it's most notable by its two flagella. So you'll see here and here. So that's one way that people recognize it. It is an autotroph, so it has a chloroplast. And you can see here in this electron micrograph, it has stored starch granules, so that's how it stores its nutrition. It does have a cell wall, and then here are where the two flagella would attach. So Chlamydomonas tends to be the model organism for single cellular green algae, and is one of those that gets used often to study photosynthesis and motility in green algae. So this is a light micrograph of Chlamydomonas, and you can see there are several of them here. This is using the 100x objective on the microscope, so you know that this is a very tiny organism. And so one of the things that can be hard to see on the microscope are the two flagella. You can see those right here, for instance. In this particular image, the Chlamydomonas has been stained, so that's why it's pink. It's not naturally a pink color. This just helps to increase the contrast so it's easier to see under the microscope.
So this is the life cycle of our Chlamydomonas. Like I said, it's a haploid life cycle. So most of its life is spent in the haploid stage. So let's just kind of go through this for a moment. All right, let's start with our zygospore. Oh, hang on, I lost my ability to point. Where is it? Oh, I see it. Okay. Here we go with our zygospore in its diploid stage. Then goes through meiosis to produce zoospores, which are haploid, and that grows into an adult Chlamydomonas. And then it goes through asexual reproduction, which is its predominant method, I do believe. So what happens is it asexually reproduces, producing daughter cells, and then each daughter cell then grows to be an adult Chlamydomonas. Now, it can also produce gametes, as you see right here, and then the gametes pair together through fertilization to produce a zygote, which then produces zygospores. So this is its sexual reproduction. So it can do asexual or sexual. Now, one thing I do want to talk about is just when you look at the Chlamydomonas, there are a few things that are very, predominant in its body. One is this eye spot right here. It's a pigment. It really does work to help it see where the most light is, so it will use its flagella to move to an area of higher light concentration since it is a photosynthetic organism. Of course, it has a nucleus and a nucleolus because it's a eukaryotic cell. And then it has what's called a pyrenoid right here. And that's where it stores its starch grains and its excess food.
The second structure type is colonial, and what that means is there's a bunch of different organisms living together, basically in like a colony, just like it sounds. And our example of that is Volvox, which is another green algae. And the first time I saw this, I think I was in middle school, and I got to see it under the microscope. And even since then, they have been some of the coolest algae I've ever seen. I still think they're really neat. So each one of these colonies, so each one of these spheres that you see over here is made up of between 1,000 and 3,000 individual cells. So there are two different types of cell that makes up these colonies. The outer portion here is made up of what we call vegetative cells. And vegetative cells are those that just, they are autotrophic. They just continue on with the daily life of these algae. But then there are cells on the inside that reproduce, and those cells are called the daughter colonies or the daughter cells. And so as those daughter cells keep reproducing, they produce their own colony that will eventually be separate from the original colony. So one thing I didn't say is of these vegetative cells, all of them have a flagella, and they work in concert to move the whole Volvox colony in one direction or another, for instance, toward light so that they can produce more sugar. So the link here to this video is very cool. It's once the daughter colony grows to a point where it gets released from the parent colony. So hop over to this YouTube site and take a look at that.
Okay, so those two structural types were all in the chlorophytes. So now we're going to move over to the charophytes and look at their structure types. So that third structural form is the filamentous form, and our example for that is Spirogyra. This is what it looks like. You have probably seen it on ponds. It kind of has that stringy hair type look to it. But they are long chains of filamentous cells, and so each cylinder contains this chloroplast that is spiral shaped. So it makes it very unique when you're looking at it under the microscope. It's very distinctive, this spiral type of chloroplast. And what happens is the Spirogyra, when they are undergoing photosynthesis, of course, release oxygen, and they keep some of that inside their cell, and that helps them to float to the top of the water, which of course is where most of the sunlight occurs, and so that's beneficial for them when they are trying to photosynthesize. So this is an unbranched charophyte, so just long cylindrical filament of Spirogyra. So here's just a look at the cell anatomy of the Spirogyra, and even more specifically, how they reproduce. It's very fascinating, actually. So, okay, let's start with the left. So the chloroplast in a spiral shape, just like we've talked about. Of course, it has a nucleus as well because it's a eukaryotic cell. They do also have small pyrenoids. Remember what we saw those in the Chlamydomonas? That's where they store their excess starch. So what happens for their reproduction? They reproduce by conjugation. So we have two filaments, 1 and 2, and they start to form these channels across between the two cells of the filaments, and literally the whole content of one filament is transferred to the other, and that forms a diploid cell because there is a fusion of the nuclei as well. And then in the spring, they undergo meiosis and they go back to a haploid filament. So I wanted to show this to you. This is an algal bloom. You've probably heard of algal blooms before. It's usually when there's an excess of nutrients dumped into waterways. Could be because of a spill, any number of reasons. But when that happens, then the algae tend to over-reproduce, and that's what we call algal blooms. The one on the right makes me laugh. But these algal blooms are fine at first, but the problem with them is that as they grow and they use up the nutrients in the water, they tend to cover the top of the water, and then they deplete the bottom layers of the water from not only nutrients but also of oxygen, because all of the algae is at the top and that oxygen is released into the air and tends to not be released into the water column, and also that blocks the sunlight. So any photoautotrophs that were below that are then cut off from their sun supply and therefore don't produce oxygen for the rest of the water column. Okay, the fourth form is the multicellular form, and our examples of that are ulva and chara. Ulva, the one on the top, is called Ulva lactuca. If you've ever seen it, that its common name is sea lettuce, and it really looks just like that. It looks like some romaine sea lettuce, but very thin. So they usually are, even though they're multicellular, the body of the organism tends to be just a few cells thick. So it's usually not massive amounts. The one in the bottom, chara, is sort of a branched type of multicellular. This is the one that is most closely related to our land plants. Remember we talked about the charophytes are the most closely related to them. This one specifically is the one that points to that. So that finishes our green algae that are in the supergroup Archaeoplastids. What else is in the supergroup Archaeoplastid? The red algae. So just to kind of show you what that looks like, first of all, we just have to mention that our red algae is basically the seaweeds, and they are of a commercial importance to us. The porphyra is the seaweed used in sushi, so if you eat sushi, you should be happy with the Archaeoplastid supergroup. Gracilaria is another red algae, and it is used, when ground up, it's used for making agar in our petri dishes. So if you'll remember, we did our bacteria swabs and we put them on agar plates. So thank you to Gracilaria to help us with our agar plates. Chondrus, another one, also commercially helpful to us because it makes carrageenan, which is a thickening agent used in all kinds of commercial products, anything from toothpaste to ice cream. Okay, so that was our red algae. So we've covered our Archaeoplastids, the red algae and the green algae Those are the protists that are in archaeoplastids. And we're just going to quickly go through chromoviolata, which is our brown algae, and then also our diatoms, dinoflagellates, and alveolates. So we're going to finish our talk today of algae by going into the supergroup chromoviolata. And specifically, we are going to look at the subheading of that called stramenopiles. Stramenopiles, that word basically means straw or hair that's thrown down like bedding. And so today we are going to be looking at the brown algae that's in this particular group. We're going to hold off and talk about diatoms at the next lecture, but for now we're going to focus on the brown algae. So brown algae includes our seaweeds, and our of which kelp is the largest, and the largest algae usually having about 30 meters in length. If you've ever been to the coast of California, you may have seen lots of kelp beds. I unfortunately have not had the opportunity to do that, but I actually have had a student from California, and it was so much fun to talk to him and his experience with that environment. It was really cool. Okay, so let's look at the anatomy of our brown algae. There is the blade. Remember, we can't actually call it the leaf because it doesn't have vascular tissue. So hopefully this won't confuse you by having it there, but what I mean is that blade of the algae. And each one of those blades has an air bladder, and that helps the leaf to stay afloat on the surface. So again, to be able to be closer to the sunlight, because even though they're brown, they are still photosynthetic. The reason they're brown is because they have accessory pigments that pick up different wavelengths of light, and that is still used in order to photosynthesize. So you can actually see these air bladders on the example of the sargassum up there. So here are some of those. And what I learned from my student is that you can pop them. They're actually, they remind me of the, that plastic sheeting that you will mail things in that has all those little air bubbles and everybody likes to pop them. This is very similar to that. They have what's called a stipe. Again, it can't be called a stem because it has no vascular tissue, but it is sort of what looks like a stem. It's called a stipe. And then it has the holdfast. And that's what anchors the kelp to rocks underneath, because on the California coast, instead of having a sandy shoreline, it has a rocky shoreline, so it has something to actually hold on to, so that holdfast allows for that. So here are some of the various algae, brown algae, that we see, and fucus, this one here, let me see if I can get to it. This one right here, that is the one that we are going to be looking at in lab. That is kelp. Now, we did mention that off the coast of California, and in other places too, I'm just saying that because it's the most familiar to us, they have what's called kelp forests, and this is a very important ecosystem, and we might talk a little bit more about that when we get into our ecology unit. But this is where many fish come to lay their eggs. So there are usually lots of baby fish. It's kind of like a nursery sort of for the Californian coast. So very, very high in biodiversity and very important. So that's one of the areas that people make sure to protect, because without that, then all of our fisheries go downhill fast. So that is all for our algae today, which are, again, our protists that are photoautotrophic. That's what they fall under. So this was our beginning introduction to protists, and we will continue next time with the rest of our supergroups.